is Zaidan, I'm a consultant medical genetics at Faisal Specialist Hospital. I'm talking to you today about replication, DNA replication and repair. This is my goal from this lecture, to describe the step of replication and to understand the mechanism of repair and recognize the importance of DNA repair in relationship with health. Okay, let's start with this. How does the presence of an organism continues? Reproduction. Excellent. Okay, how genetic information is transmitted from one generation to the next? Replication, because if we don't replicate this material, it will vanish with time. How does the extra DNA come from? We replicate, so we need material to replicate the DNA, isn't it? So this DNA, from where we get it?
It's pre-existing within our cells. It's pre-existent within our cells.
كل حاجة تخلص. Pre-existing will finish. Recycling, as you said, recycling, but also there is de novo, something from.
Doctor, we can't hear you.
So either de novo synthesis or nucleotide salvage. What is the mechanisms for all live cells to give offspring?
The mic is not working. Nobody can hear you at the back.
Sorry. Sorry about that. So, what is the mechanism for all live cells to give offspring? Now we are talking about on the level of cells.
Division.
Division. Cell division, excellent. Okay. When does a cell divide? So the cell will divide to keep the species. What is the time of the division? What is the condition that need to be fulfilled before division? When all the material in this cell is double. Okay. When the DNA content has double, division will take place. طبعا the division will depend on the type of cells. Some cells will just stop in the G1 stage. You have been through the cell cycle and Dr. Hassan. So like neurons, they stop. This is why when we have a problem with neurology or CNS, it's very difficult because the repair or let's say replacement is very difficult. Very fast in skin, very fast in GI cells, liver cells, but very, very slow. Sometimes it's arrested in CNS and bone. Okay, so double the material before division of the cells. This is the structure of DNA that Dr. Hassan described with you. It's composed from nitrogen, base, phosphated group attached with a sugar or deoxyribose. This is also just illustration. And this is the life cycle, or the cell cycle, sorry. The interface, which will compose of 80% of the cell cycle. And you can see it here. This is the interface composed of GAB1 or G1 phase, which is the routine situation of the cell. Its function normally takes over the ordinary stage of the cell. Prior to division, it has to synthesize or replicate the genetic material, synthesize the needed protein, needed organelle, so it will go to the S phase, followed by G2 phase, which will continue, completing the required material like protein, duplication of several cell organelles and protein. But S phase is mainly of the DNA and genetic material. Then followed by M phase, where the division happens. DNA replication is semi-conservative. It means that the maternal strand will be replicated, exactly similar. So you keep one of the strands as a maternal, and you create another one as a paternal. This is what happened with multiple replication. Let's talk about the speed of DNA replication. The speed of DNA replication in the human is about 50 nucleotides per second. So imagine that we have a chromosome composed of 250 million base pair, and we want this chromosome, short chromosome, to be replicated. What do you think the time for this process? I calculated it here. It is 58 days. Do you think this is what happened? What is happening? In 58 days to replicate only one small chromosome?
No.
Will take 2 months? No. This is not. What happened? That it will take only a few hours. Why? Because the replication will happen in parallel, with multiple replication origin. So it's not like this one replication origin and you go from that. No, there is multiple. It's exactly similar to the concept of parallel and serial in the computer. So if you have it parallel, you will do it faster. Okay, the step of DNA replication, let me start from the final step, then we go back to the details. So you can imagine what I'm trying to say. Yeah, this is it. This is the double strand that we need to replicate. First of all, we need to separate it. Then, this is the separation, the first step. Now we need to replicate. To start replication, we need something known as RNA primer. زي منصة الإطلاق. Polymerase enzyme, polymerase 3, which is responsible to build up nucleotide one by one, always from 5 direction to 3 direction, always. Work in this direction. We need a small RNA primer here that polymerase 3 will attach to this primer and start replicating one by one from 5' to 3'. It's exactly opposite to the template, maternal template. So we need one primer here. Then it is a continuous process from here. So the first step is separation. The second step is primer, which is here and here. The third step is elongation. Now you put the primer, you start replicating. You need to extend the replication strand. This is the third step, elongation. Fourth step, you need to remove this template. You don't need it anymore because it's RNA and this is DNA. You have to remove it by another enzyme known as polymerase 1. Then you remove it and you replace it by DNA. Finally, you have to ligate it. Ligate the new small piece with the old piece. You have to notice that in this direction you have no problem. It's one primer is enough to take care of the whole process. But in the other strand, this is known as leading strand. This one known as lagging strand. Because assume that we separate, we put the primer here, okay? The replication will start in this direction, okay? When you separate further, you need another primer here. Because the replication, as you said, the polymerase will not work in this direction, which is 3 to 5. It will work always 5 to 3, which is 5 to 3. This is why we have multiple primers. Every now and then we need another new primers. When we separate new primers, this is lagging strand. Lagging strand. Okay? This is 5 steps of replication in summary. and this can summarize the whole process. Helicase, then the enzyme known, the enzyme which separate is known as helicase. When you open by helicase, there is tendency for these two recently separated strands to go back. So you need something to stabilize them, which is single strand binding protein, which will come here and stabilize these two, not to reannihilate. This is the first step. So helicase enzyme, unwind, SSP, stabilize, is the first step. Second step, RNA primase enzyme, which will insert RNA primer here and here. And this one will continue after the RNA primer. The third step is elongation by an enzyme known as polymerase 3, which will work always from 5 to 3 direction. This is why it will work as segments in the lagging strand, while it will work as continuous in the leading strand. This is the third stage. The first step, you don't need these anymore. You have to remove them by an enzyme known as polymerase 1, which will remove them from the same direction from 5 to 3. Then has another function to put a DNA instead of them, always from the same direction from 5 to 3. Now polymerase 3, the one which will polymerize and replicate, and polymerase 1, which will remove the RNA and put DNA instead of it, both of them, they have another capability to work on reverse direction for something known as proofreading. Proofreading, there is a mismatch, a mismatched nucleotide incorporated during the replication. These two enzymes able to go back one step and remove this mismatch and put the right DNA nucleotide, the right nucleotide. So these are the steps. Let's go. And now you have the big picture. Maybe it will make it easier to follow the steps. So we said the first step is separation of the two strands and formation of replication fork. We talk about single helicase, which is the enzyme which will open. We talk about single strand DNA binding protein, which will stabilize the two recently separated. DNA A protein, what do you think? Who knows what is the role of DNA A protein? I put it here. Who can guess? Okay, DNA A protein will go and target the weakest area of the double strand. That the helicase will go and start unwinding. The weakest area is always AT-rich area. AT because there is 2 bonds, while CG, 3 bonds. So this one will target the area which is AT-rich, and the process will start in these areas. Supercoil problem. After unwind this, template, after a while there will be a problem with supercoil. Say if you have a rope and try to open it, unwind it, ultimately the rest of the rope will flip over itself. And this is supercoil. And you need to release this tension, otherwise you stop. The helicase will not be able to open further because there is a tension. Clear? Clear?
Yes.
Okay. So we have 2 enzymes known as topoisomerase. Their role is just to remove this tension down toward the template. This is just a description of the process taken from a lecture from my colleague Dr. Hamad. We're giving this lecture together, and we decided to merge together, so it will be we cover the same information for both. But now I left it. I'm giving, now I'm the only one who gave the lecture, but kept it because it's descriptive, more descriptive. If you need to remember what I've said, you can read it. This is the SSP. This is the sobor code issue. You have type 1, which will cut one strand only and flip this through this, small nick. Then you will seal it. With time, every time and every now and then, you have to topoisomerase will cut. You flip back the rest of the strand to remove the tension, and you continue opening and unwinding. Here, topoisomerase 2 will cut both strand and you flip it over each other and you remove the tension. The second step, initiation of replication RNA primer. We said, we described that. We'll end up with two strands, maternal strand, or two maternal strands, and we build two newly synthesized strands. One of them is leading, continuous. One of them is lagging, comes in segments. Okay, this is the description. This is what I've said. In the lagging strand, it's continuous. It's interrupted. RNA, replication. RNA, replication. RNA, replication. Then the RNA will be removed and replaced by DNA. Here, leading strand, because it is 3 to 5. And the replication will go from 5 to 3, which is continuous process. But here, it is 5 to 3, so the replication flow is opposite, 3 to 5. It has to be interrupted. Clear? Is it clear? Third step is elongation of DNA. We described that, polymerase 3. Very highly processive, يعني عنده شغلات كثيرة يسويها. There is a pyrophosphate release, so it's energy. There is a needed energy in this process. It will polymerize from 5 to 3, and it will stop if any nucleotide is depleted. Common sense. If you don't have for example, adenosine, and now there is in the template, maternal template, there is T. And now you need to incorporate adenosine. You don't have adenosine, so it will stop. The process will stop. Proofreading, we described that. There is a process, there is capability to go back one step and remove the mismatch nucleotide and replace it with the correct one. This is how polymerase work from 5 to 3 5. Always. Okay. Same, including fragmented, lagging strand, fragmented RNA primer, polymerization, RNA primer, polymerization. Further annealing, RNA primer, polymerization. Here, it's only one. It's a continuous process, leading strand. SSB, helicase, elongation. Proofreading, this is the normal situation. If you have a mismatch, the same enzyme will go back, remove the mismatch nucleotide and replace it with a new correct one. Elongation. Fourth step, excision of RNA primer and replacing it by DNA. We said DNA polymerase 1. We go to the RNA primer and remove it from 5 to 3 direction. Remove. Then go back and insert DNA. Again, from 5 to 3 direction. If there is any mismatch nucleotide, it will go back and remove the mismatch, put the correct one, and continue. So 3 function. The last step, we have multiple segments, especially in the lagging strand. We need to ligate them together. And this is the function of DNA ligase. Also, energy is required. That's it. This is the This is the ligase. This is summary, simplified summary of the process. Yeah. Now, when you reach to the right end, the last few nucleotides in this chromosome, you use to put RNA primers and use it to do the replication. Now, if you put the primer in this area, خلاص, this is the last few nucleotides. If you put the primer, you cannot replicate them, and you cannot replicate without a primer. So, how this will be solved? This will be solved by an enzyme known as telomerase. Telomere is the right end of the chromosome, صح? So this is telomerase will create an extension to the maternal strand that will be used as a place for the RNA. That will be used for replication. Then all these will be removed. This is the last few nucleotides that need to be replicated. If you put the primer here, you cannot replicate them. So the telomerase will build up more few nucleotides further. That will be used as a template for the primer. Then the RNA, the DNA polymerase D will attach and start replicating. After finishing this process, all this will be removed. Clear? Review, rapid review. Helicase, the function for leading and lagging strand. The helicase function is what?
Unwind.
Unwind. Excellent. Single strand binding protein. Stabilize from rewinding again. Double isomerase. Supercoil issue. Solving the supercoil issue. Primase. RNA primers, synthetic RNA primers. بس هنا لحظة. There is a difference now between the function in the leading strand or lagging strand. What's the difference for the RNA primase? Excellent. Only one leading, and it is multiple in the lagging. But DNA polymerase 3. Well, this is okay. DNA polymerase 3. Elongation. Elongation. Continuous elongation in the leading and
in the lagging it's
fragmented elongation. Sorry. Polymerase 1. Remove primers and replace by DNA. Multiple lagging and once leading. DNA ligase.
Seeding strand.
Ligate the newly synthesized segment, newly synthesized multiple segment in lagging, and newly synthesized segment in leading. Joint with exactly. Okay. When I was at your level, I always ask myself, why do we need to bother ourselves with these details, very dry information that what I will do, I will give you some medical application of DNA replication so you can understand why we want to understand this very well. And we know that we appreciate that this information is translated to treatment, to intervention, to medical care. I have 2 points. Reverse transcriptase. Do you know AIDS, HIV virus?
Yes.
HIV virus used a reverse transcriptase. So the RNA, it's a retrovirus, which means that the DNA, the genetic material in this virus is RNA, and to infect a human cell, which is DNA, this virus has to create a DNA which carry the same genetic information of its own. So this is an enzyme will create DNA taken from the RNA of the virus, known as reverse, because it's the opposite. The process is opposite to what we know. Then after the DNA is created, it will integrate it in the human cell, which is infection. This is the infection. How this virus infected the cell, it introduced its genetic material in that cell. This information is important because to understand this, this will help you to defeat the HIV virus. And I'll show you now. Nucleoside analog, you remember we said if there is depleted nucleotide, the process of replication will stop. صح? Assume that it is not depleted, but this nucleotide is cheated. We do some modification that the polymerase will take it and attach it to the newly synthesized strand, then it cannot do anything further. يعني مغشوشة. Analog to any one of the nucleosides. And this is the concept of multiple medications. Example of this is anti-cancer and antiviral medication. I'll show you now. What is acyclovir? Have you been through this ما سمعتوه أيام الكورونا? It's antiviral medication. Okay, zidovudine is another antiviral used for HIV. Gemcitabine, it's a treatment for cancer, pancreatic and lung cancer. Okay. The idea of this, I'll take acyclovir. Acyclovir is a guanosine analog. So similar to a guanosine, the polymerase will take this guanosine, this acyclovir, which is very similar to guanosine, thinking that this is guanosine, and will attach it to the newly synthesized. Then after this step, the process stops. The polymerase will not be able to attach anything after that. So you stop the replication of this virus. يعني we cheated the mechanism. We cheated this process. The difference is very simple. This is the guanosine, sugar, phosphate with hydroxyl group, and base. We just remove the sugar residue, which will contain the hydroxyl group, which is the area that the next nucleotide attached to, the phosphodiester bond. Attach hydroxyl. And then we put the same, but we remove just the sugar with the hydroxyl group. This is the acyclovir, and this is the natural substrate. Elongation, this is the usual. Hydroxyl group with the phosphate of the next nucleotide. There is no hydroxyl group, it's a dead end. When you incorporate this, which is acyclovir, in the DNA, newly synthesized strand, the process stops here. There's no further replication. Zidovudine is the same. It's a thymidine analog. The difference, the hydroxyl group was removed and we put nitrogen. Nitrogen will not be able to incorporate it with the phosphodiester bond. So you stop the process of HIV infection. Clear? This is the idea is clear ولا لا? The concept is very simple. يعني the detail يعني ما لكنه the concept to change the structure of this compound, we name it as the nucleoside analog. This is the third example, gemcitabine. Deoxycytidine, هذا natural. We change this hydrogen group to fluoride. And this will stop the replication of the DNA of the person himself. So you control the uncontrolled replication of cancer. Video clip, small, very informative, because these things, you need to see them in motion to understand the process. It's a simultaneous process, and unwinding, stabilization, primers, elongation, together. It's not like what you think in one step, in a second. It's a continuous process. So I recommend you to see it. How many horses?
4, 3, 5, 4. Where are you saying 4?
مش ناخذ اعلى عدد ولا ناخذ اقل عدد؟ الكومن سنس انه ناخذ اعلى عدد.
5, 6, 7. I see 7.
Who said 6?
I said 6, 7.
كم اعلى عدد وصلتوا؟
7.
ها؟
7.
لا عاد قوي لي سنة. لا
I said
لا
6.
5. 5. يلا تندس قلنا صح؟ بسم الله. How does familial colorectal cancer happen? But I don't expect you to tell me, but I want you to raise this question. Imagine there is families, you may find 5, 6, 10 persons in this family end up having colorectal cancer at early ages. So there's something for genetics here. You see, disease that common in this family compared to the others. Xeroderma pigmentosa, another disease, skin disease with more than 1,000-fold increased tendency to have skin cancer if exposed to sunlight. Okay. These two examples, I brought them because these two diseases is caused by defective repair mechanism, full stop. To understand how serious the repair mechanism. What do you think will be the consequences of a few mistakes? يعني خلينا نقول مثلا 5 مليار 5 مليار مثلا replication, only one mistake. What do you think will happen in the process of DNA replication? Excellent. Mutation, and this mutation will be transmitted to the next generation. If it is skip the repair. The concept here is this is, even if it is very few, it is catastrophic. Alhamdulillah, Allah created a very, very, I can't say anything in the repair mechanism. It's a multiple steps to control, to repair the mechanism, multiple layers of repair. As I told you, even the enzyme which replicate has the capability to proofread. And from the beginning, there is a process of repairing, continuous repair. Because the result is catastrophic. Assume if you allow 1 or 2 mutation in every letter, ultimately you end up with another creature. The occurrence of mutation DNA, if left unrepaired, would have serious consequences for both the individual and subsequent generation. Median gene mutation among different genes is 1 to the 10 power minus 6. There is a small risk of this mutation. Some of them will not repair. However, most of them will be repaired. Sources of damage that need to be repaired. We have either depurination of nucleotides, so the purine base will be removed, or deamination of nucleotide, the nitrogen group will be lost, or demethylation of the nucleotide. Sometimes it's a reaction with chemical mutagen in the environment. This is why we say, oh, this material is cancerogenic. Don't touch this. Don't inhale this, because they affect our DNA. Exposure to ultraviolet or ionizing radiation The xeroderma pigmentosum that you saw is a person like you. However, he missed the mechanism that protect him from ultraviolet from the sun. You spend the same time under the sun, he will develop cancer. You spend the same time, or even more, you have nothing. This is just an illustration to show you here the sources of damage to our DNA replication stress, alkylating agent, oxygen radical, and ultraviolet, ionization radiation, chemotherapeutics, polyaromatic hydrocarbons, moad material that may affect our DNA. And this is the type of effect of this material in our DNA. It can lead to base mismatch that we talked about earlier. That G to A, this is a mismatch. It has to be corrected, or C to T, so it has to be corrected. This is known as base mismatch, or single strand break. Sometimes the agent will lead to a break in the single strand. Sometimes DNA adducts or pyrimidine dimers. There will be dimers between two pyrimidine together. These are the effect on the DNA. Now the third line is the mechanisms of repair that our body will use to correct these. But there is overlap between the affecting materials, as you can see. It can lead to more than one effect on our DNA. This one can be corrected by more than one mechanism, base mismatch repair. It could be repaired by single strand break as well. So there is overlap between mechanisms. The repair usually happens in multiple steps. This is the majority. However, sometimes there is only one step. Like in bacteria, there will be, two thymidine group will adjacent to each other, they attach to each other. So this will stop the replication. And this, in the bacteria, will be resolved or repaired using the energy of ultraviolet. So the bacteria will use ultraviolet to just remove this dimer, thymidine dimer. So this is an example of direct repair. But in the majority, especially in human, there is a cleavage of the DNA strand by endonuclease, then the removal of damaged region by exonuclease, then insertion of a new bases by the DNA polymerase, similar to replication process. And you remove, you put a new one, and you like it. Okay. The repair could happen as on a level of single strand damage. Sometimes there is double strand damage. Double strand damage, you lost in the single strand, you have a strand that keep the information for you. You can replace, but the double strand, you lost both strands, you don't know what is there. You have no reference to I'll concentrate probably on this a little bit. And types of single strand is, we talk about mismatch repair, which could be base excision repair and nucleotide excision repair. From the name you can imagine, base excision repair, the mechanism will remove the base only and correct it. The nucleotide, no. You will chop a small piece, which is including multiple nucleotide, and you put a new one. Base excision repair, sometimes single nucleotide in the double helix are altered by a chemical reaction and become mismatched. Example, مثلا hydrolytic removal of cytosine amino group. When you remove this amino group from the cytosine, it will be uracil. Uracil is not a match in the DNA, so you have to remove it. So this will be removed by base excision repair, because it's only one base that is altered. This is an example. C, you remove the amino group from the cytosine, then it will be altered to uracil. But uracil is not a correct match, so you need to remove it. It will be removed by glycosylase. Then you'll end up with a pyrimidic nuclease. You need, a pyrimidic endonuclease. It's a nuclease that cut the strand, which has no pyrimidine. So this is the area that it will work with. So it will cut the strand, then you insert the correct match, which is cytosine. This is also the same fancy. Then finally, you ligate it. I'll show you more detail. But the concept, you remove the base, you cut the strand, then you remove, then you insert the correct match, which is cytosine, then you ligate. Right, okay. The second type is nucleotide excision repair. A large multi-enzyme compound scan the DNA strand for abnormalities. Then here it detects nucleotide not married, mismatch. It will be cut from here, and you remove all these nucleotides, and polymerase will insert the correct nucleotide one by one. Then finally, you need to ligate this one, similar to the polymerase. And by DNA ligase, you ligate this nick. It's the same description. Here, the example is thymidine dimer. The pair of thymidine dimer. Again, it is, you remember, thymidine dimer, we said in the bacteria, it's a direct. The bacteria will use ultraviolet to just open it, and it's a direct process. In human, no. See, we use nucleotide excision repair to correct this one. You don't have the facility, the capability to correct it by ultraviolet. So it has to be cut from here to here. You remove this one and you insert the correct one. Okay. This is examples of now this process of repair is controlled by genes. Genes control this process of repair. So if we have a mutation in the genes responsible for this repair mechanism, this can explain to you how cancer will be familiar. For common sense is cancer. We understand that cancer has nothing to do with the genetics. It's something because we are exposed to something or to cancerous genes, cancerous substance. But here, you understand how come cancer be genetic, because some genes controlling the repair mechanism, and these genes are mutated, not functioning well, so the repair mechanism is not functional. You end up with accumulating mutation, which ultimately lead to cancer. This is the answer of my question in the first slide of repair. We said how we have a familial colorectal cancer, because those families, and I saw them in reality, and I saw cases of this, they have a mutation in this MYH gene, which render this gene not functional, ultimately affected the base excision repair, so the body is unable to repair the base mismatch, and they accumulate mutation and end up with colorectal cancer. Xeroderma pigmentosum, the other example that I showed you, there's multiple genes in the Xeroderma pigmentosum genes. Any mutation in any one of them may lead to that severe disease. This is just a few points about Xeroderma pigmentosum. The most important, that more than 1,000-fold increased risk of cancer at ultraviolet exposed area. You don't need to know all these, but this is just for information. There is a failure, image recognition failure in the Xeroderma pigmentosum patient. There's a mutation. It's multiple genes. One of the genes, for example, responsible to recognition of the area of thymidine dimer, for example. If this gene is not functional, there is a failure in the recognition of the problem. There is other genes responsible for the production of this protein, which is responsible to unwind. Yani, what I'm trying to tell you that multiple genes, every one of them is responsible for one single function of this repair mechanism that the body used normally. Any defect in any gene may lead to xerodermatosm because failure of this stage, or this stage, or this stage, or this, or this. You have to protect it after that. This is another disease, premature aging, 48 years, as if she is 80. It's a problem of repair mechanism. Bloom syndrome, Werner syndrome, Zaidan, كل هذه, all these are examples of diseases caused by defective replication. This is general information just for your information. Again, this is the triggers. This is the lesions that happen because of these factors. This is the repair process that used usually to correct this problem. And these are genes, all these genes are implicated in this type of repair. يعني احنا تكلمنا عن مثلا زيروديرماتوزم دي هذا كله genes of زيروديرماتوزم. هذا نيو TYH هذا كله كانسر. mismatch repair, mismatch repair genes SH1. بس أنا this slide just to show you, there is this repair mechanism controlled by multiple genes. Any defect in any of these genes may lead to cancer. This Nobel Prize Chemistry 2015 was awarded to Thomas Lindahl, Paul Mordech, and Aziz Sanka for having mapped how cells repair damaged DNA. Nobel Prize عشان repair. It's very important. طيب, thank you very much. Let me have another